Hello and welcome to the short workshop in this afternoon. So I'd like to introduce the authors. So this workshop will be given by Mikhail Dosmorov and Spiro Stilianoudakis from the Virginia Commonwealth University in Richmond, the US. So very welcome to the workshop. And now I hand over to Mikhail and Spiro. Thanks for being here.
All right. Thank you, Simon, for the introduction, and thank you, everyone, for coming. We are in Richmond, Virginia, from Virginia Commonwealth University. We are interested in analyzing three-dimensional structure of the genome, developed several bioconductor packages for this purpose: Hi-C compare, multi-Hi-C compare, spectral TAD, and TAD compare. And this workshop will demonstrate our latest work, which will be presented by Spiro. So Spiro, please go ahead.
Okay, so thank you to Simone for the introduction. Thank you, Dr. Dosmorov. And thank you, everyone, for attending this workshop. This workshop's titled PreciseTAD, a machine learning framework for precise 3D domain boundary prediction at base level resolution. So essentially, this workshop will be going through the functionality of this PreciseTAD R package that Dr. Dosmorov and myself have been working on and submitted to Bioconductor, and it's been accepted recently. So briefly, I want to go over kind of the overview of this workshop and show some key resources. So first, I'll be going over some background into the 3D structure of the human genome and some motivation of why we created this R package and the research that went along with it. Those will be found in these slides through this link, which we'll go over in a second. Next is the link to the actual GitHub repository that's associated with this workshop, which is this page you're looking at. And the next is the Docker image that you can use to work on the core of the workshop that we'll get into later. You can either build the Docker container using these commands, or you can go to the actual cancer data SCI site and then launch the workshop from there as well. And then lastly, the package down site that's associated with this workshop, which we'll use a little later on. Okay, so diving into the slides I have presented here. So again, these are just a repeat of the resources provided that I just went over. Okay, so a little bit of background into the 3D structure of the human genome. The human genome is actually relatively large. It's about 3.2 billion base pairs. And in order for it to fit inside the nucleus of a cell, we know it has to form these compact loops and folds. And this looping and folding actually, it doesn't occur at random. It's actually organized at multiple scales. And so kind of at the most finest scale, at about the 10 kb scale, we know DNA wraps around histone octamers and forms nucleosomes. However, zooming a little bit further out into the 10 kb to 800 kb range, we see that chromatin forms these regions of highly interacting DNA known as topologically associating domains, or TADs for short. TADs themselves, if you zoom out even further, actually organize into what is known as AB compartments, which refer to active and inactive chromatin. And then zooming out the furthest, we see chromosomes take their kind of distinct chromosomal territories and make up what is known as a fractal globule that fits inside the nucleus of a cell. So the purposes of our research are focusing on that intermediate scale, these topologically associating domains, or TADs. Now, excuse me, why do we want to study TADs? Well, they've been shown to be stable in dynamic structures. By stability, I mean they've been shown to be highly conserved across species and cell lines. And by dynamic, it's been shown that they've been associated with gene regulation. And actually disrupting the boundaries that demarcate TADs, it's been shown to lead to developmental disorders, rare diseases, and even cancer. So understanding kind of the mechanisms that lead to the formation of these TADs, these regions of highly interacting chromatin, is a interesting area of research, especially in the field of genomics. So we can visualize where TADs are along the linear genome by looking at what is known as a Hi-C contact matrix. This is sort of a byproduct of Hi-C sequencing. And so essentially a contact matrix is just a square and symmetric matrix that measures the amount of contact that occurs between any two regions on the linear genome. And these regions are controlled by the Hi-C data resolution. Resolution controls how much you bin this contact matrix along the X and Y axes. And again, that resolution is controlled by the sequencing depth. So if you perform Hi-C sequencing at, say, 50 kb, you're binding the linear genome every 50,000 base pairs, and you're enumerating how many contacts lie within any two regions IJ of 50 kb dimension. So as you can see here, as you can probably surmise, red regions correspond to higher levels of interaction, white regions correspond to lower levels of interaction, and then TADs themselves form along the diagonal as these triangular regions, or if you think about the symmetricness of the contact matrix, these square regions along the diagonal. So there have been tools proposed recently called TAD callers that simply query a contact matrix and attempt to find the boundaries that demarcate these TADs along the diagonal. So an example of kind of what the DNA might look like in 2D space is shown here, and so a TAD caller would try to query this contact matrix, which is just count data essentially, and attempt to determine the boundaries that demarcate these two TADs. So a good TAD caller would identify these boundaries as being TAD boundaries, and so we also know that TADs themselves are sometimes hierarchical in nature. So you could have a situation where you have a meta TAD and then two subTADs within them. Now that's easier said than done because there are a lot of pitfalls that are associated with TAD callers. One is that they're heavily reliant on that Hi-C data resolution. So the boundaries that a TAD caller, conventional TAD callers, identify as being potential TAD boundaries have to be divisible by the resolution. And so this resolution spans a wide, it has high variability. So you can have Hi-C sequencing anywhere from 250 kb all the way down to 1 kb. So your results can be highly variable. Secondly, and more importantly, they ignore a lot of potential signal, namely the role that the role that functional genomic data or functional genomic elements have on chromatin folding. For instance, we know from the literature that certain transcription factors are enriched at TAD boundaries or colocalized with other transcription factors at TAD boundaries, namely CTCF, RAD21, SMC3. These are just a few examples. Those transcription factors are components of what is known as cohesin, which contributes to this looping formation that leads to TADs. So kind of the primary research goal here is can we improve. First, can we transform this problem into a machine learning problem? And can we leverage these genomic elements in a predictive model to more precisely predict these TAD boundaries? And are our predicted boundaries more biologically meaningful than a conventional TAD caller, in essence? So getting into just how we formulated this problem, again, we want to transform this into a machine learning framework. And in our case, we're going to be working with binary classification. So as you may know, two components of predictive model, specifically binary classification model, you need your response vector and you need your feature space. So to construct our response vector, we took a conventional TAD caller, a popular TAD caller called Arrowhead, which I'll discuss in a little more detail in the body of the workshop. So we took a TAD caller, Arrowhead, we called TAD boundaries, and then we binned the linear genome according to whatever resolution we performed the TAD calling. So for instance, if this was performed at 5 kb resolution, we binned the genome every 5,000 base pairs, and then we labeled it a genomic bin or a region as being a TAD boundary region if it overlapped with this ground truth boundary. So as you can see offhand just from this toy example, you're going to end up with heavily imbalanced data. So a proportionally high number of non-TAD boundary regions versus boundary regions. which can be detrimental to most machine learning algorithms. So to correct for that, we opted for simple random undersampling to create balanced classes. Now that's how we would construct the response vector. So if you think about rotating this 90 degrees, that's your response vector y. Now to identify the second component, the feature space of our model, we took a series of genomic elements. These are essentially ChIP-seq data. So each ChIP-seq data, for example, transcription factor binding site, has its own chromosomal start and end coordinates throughout the linear genome. So for instance, if we're working with a particular element, X sub I, it has start and end coordinates represented by these green regions. So to create its specific feature vector, X sub I, we enumerated the distance between each genomic bin to the nearest ChIP-seq peak region of interest. And so for however many elements you consider in your model, you would have that many feature vectors. So in this case, you would have P feature vectors for each genomic element. And so that composed our data matrix that we used for modeling. Now, the purpose of this model is essentially to predict TAD boundary regions. So that's not as precise as we want to get. We want to get to an actual boundary point at base level resolution. So that's where the crux of this package comes in, the preciseTAD function, which the package is named after. And so essentially what the preciseTAD function does is it leverages a predictive model. In our case, we're working with a random forest. And it works in conjunction with dbScan, a density-based spatial clustering, and partitioning around medoids to essentially predict and choose the most biologically meaningful boundary among a set of potential TAD boundaries. And so how that's done is sort of illustrated here in this pseudocode. So essentially you take a random forest model, a TAD boundary region prediction model, essentially, conducted by random forest, although it's, as I'll discuss later, it's up to the user what model they want to use here. And then for each chromosome that you want to make boundary predictions on, you do the following. You construct a base level resolution predictor space called A, which has dimensions n by p. Now this is similar to how the feature space was constructed, but it's done at a base level resolution. So n here is the total number of coordinates for chromosome I. P is the number of predictors that you're using, number of features. You assign a probability threshold T and an epsilon neighborhood epsilon. This corresponds to a parameter in dbScan. And then you evaluate your model on A, and you get the probability vector pi sub n of each boundary's potential, of each base coordinate being a potential TAD boundary. You subset that prediction vector according to whatever your probability threshold is, T, and we opt for this in order to avoid potentially inflating the number of boundaries that we're predicting. And so once you subset your probability vector, you create a dissimilarity matrix, which in this case is simply a distance matrix, a pairwise distance matrix of the subsetted probability vector. And then you apply dbScan on your distance matrix D. You assign min points to be 3. That's another parameter in dbScan. We assign it to be the default 3, and then your epsilon neighborhood that you opted for in step 4 here. Then within each cluster identified by dbScan, you perform PAM partitioning to identify the cluster medoid within each of these clusters. This cluster medoid is what we call our preciseTAD boundary point. That's our boundary coordinate, our TAD boundary coordinate that we identify using our algorithm. Now, this method also comes along with a sort of a proxy for a confidence interval, if you will, and that's what we call a preciseTAD boundary region. So for each boundary point, a region is assigned as the region that's the genomic coordinates that span each cluster, essentially. So that's what we call a preciseTAD boundary region. So that's just a little bit of a background into how the algorithm works. And so what I'll also be going through in this workshop is how we evaluate our boundaries and how we compare them to eventually we'll compare them to Arrowhead, the tag caller I mentioned before. And to do that, we looked at several things. First, we looked at enrichment heat maps and signal profile plots. These are essentially two tools that can evaluate how enriched known molecular drivers of 3D chromatin are. For instance, how enriched is CTCF around your boundaries. We know from the literature that you should have high levels of enrichment of CTCF near your boundaries. And then statistically speaking, are our boundaries closer to these, say, CTCF sites than Arrowhead? And then as a way to evaluate conservation across cell lines, we'll be looking at Venn diagrams and then corresponding Jacquard indices to evaluate how the strength and the overlap between two cell lines. So that's kind of a breakdown of how PreciseTAD works and how we will be evaluating and comparing it. Again, this is just reiterating the workshop resources. And so, like I said, going back to this GitHub repository, these slides can be found here if you want to look through those again. Okay, so moving on to the actual workshop, just as a demonstration, we'll go to the package down site, which you can view here. So we'll open that here and go to the actual vignette. So I just want to ask, is that zoomed in enough for everybody? Potentially I can zoom in a little more. Okay, so now we'll get into actual hands-on implementation of PreciseTAD. So again, if you want to follow along, you can launch the workshop through this cancer data SCI website. I've already launched it and ran some things which take a little bit of time, so hopefully I can avoid waiting on that. So going back to the package down site. So as always, when you're working with R, there are sometimes a situation where you have to load in libraries. So that's what this step is. We load in the primary package here, PreciseTAD. We have an additional package called PreciseTAD Hub. This is an experiment hub package or an experiment data package. If we have time, I'll talk about it. You don't need it necessarily for this workshop, but it's got some supplementary models that we've stored on the cloud there. That might save you some time if you want to implement this on your own. Okay. So I created this workshop with the intent that if you wanted to go and work on this on your own, there's plenty of detail and you shouldn't get stuck with anything. So there's a lot of detail here that I won't read word for word. But the primary goals of this workshop is, one, to show that PreciseTab predictions are more enriched for these known molecular drivers of 3D chromatin. Secondly, PreciseTab boundaries are more conserved across cell lines, which we know is a characteristic of TADs. And then thirdly, using the methods developed, we can actually train models on one cell line to accurately predict boundaries on another cell line. If, say, for instance, a cell line doesn't have high resolution, high C data available to it. Okay, so first, I want to kind of detail how we're obtaining our ground truth TAD boundaries that we're using to construct our response vector. Again, we're using the Arrowhead tag caller. This is a tag caller developed by Aiden Lab. I have embedded links here that you can read more about Arrowhead, and then where the corresponding high C files are stored on Geo that need to be used with Arrowhead. And then again, you can kind of read the full protocol of how these high C files are derived or created in this embedded link as well. But essentially, Arrowhead is a Java tool where you take your high C files. In this case, we're working with kind of a combined set of replicates. I think 18 separate replicates. You can read more about that at this source. And we're implementing Arrowhead at specific resolutions up top here, and then for each chromosome. And so with Arrowhead, you have to perform this per chromosome. That's what this loop is, and then at a specific resolution. So this kind of example code is performing a bunch of Arrowhead at all the autosomal chromosomes and then across a variety of resolutions. And as an example of what that looks like, I have shown that here. So the output essentially gives you the chromosome, the TADs, the start coordinate of the TAD, and the end coordinate of the TAD. Again, since TADs are symmetric across the diagonal, these next three columns are identical. So essentially just these first three columns are what you need. But we've provided the Arrowhead and the output of Arrowhead performed at 5 kb in this workshop. So, I'm sorry, in PreciseTAD. So if you load that data, you can see what that looks like, and it's stored here. So we're performing this example for this workshop at 5 kb for GM12878 cell line. So just hold on to that for now. We'll have to use that later on. But next we'll get into obtaining the ChIP-seq data that we'll use to construct the feature space. So to do that, we've compiled a bunch of different transcription factor binding sites from ENCODE. You can find those here from the ChIP-seq matrix portal by ENCODE, but again, I've provided a list of 43 separate cell line specific transcription factor binding sites in this location of this workshop. Again, you can find that here. Okay, so those are stored here as bed files. We talked about this in the slides, so I'll skip that. So now getting to the actual model building step, combining again the response vector and the feature space. There's a little bit of a kind of pre-processing that you have to do before you combine those components. So the first function is this extract boundaries function. Again, it takes the data frame or matrix of your TADs, which in this case we're using Arrowhead, and it turns it into a GRanges object. So you go from each row being a TAD, how many coordinates it spanned, to each row here being the boundary that Arrowhead calls. We set preprocess to be false. Preprocess controls whether or not you want to filter out TADs that exceed 2 megabases. In this case, we're not interested in doing that. The chromosomes that you want to extract, we're working with 1 through 22, and then the resolution that you want to work with that corresponds to essentially what TAD color was performed at. So this will be used to construct the response vector. To get the feature space in the format, we need to transform each of those bed files into a GRangesList object. So to do that, you can take advantage of the bed to GRangesList function. Again, this is in PreciseTAD as well. You provide the path that you want. Now, if you are working along with me, I know I'm going a little fast. I'm sorry, I'm running out of time. I want to fit in as much as I can here. If you're working along with me, this path you probably need to change to, I think, home/RStudio, and then this. And you can simply read in the bed files that have been stored in that path. And so again, it takes each bed file and it transforms it into a GRangesList object. And if you view the names, you can see just the full list of transcription factor binding sites that we're working with here. So there's 43 in total. Okay, so again, we've now set up the component that we need to create the response vector and the component that we need for the feature space, and we can meld those two now using the CreateTagData function. So we specify what ground truth boundaries we're using. We specify the resolution that we're working with. In other words, how big of bins to bin the linear genome. So we're opting for 5,000. And then the list of features to consider, essentially. We specify what type of feature space we want. For this workshop, we're working with distance, as I illustrated in the slides. There are other options available in the CreateTagData function, which you can read more about later at your discretion. And then again, we specify what type of resampling, if we want any, to balance the classes for the response vector. Again, there are other options for resampling here, which you can read about. And then for the training chromosome, this specifies essentially what data are we restricting for the model to be trained on, and what data do we restrict to test on. So the convention that we have worked with is that we restrict whatever chromosomes we want to make predictions on, or whatever chromosome we want to predict TAD boundaries on. We restrict that for testing, and so we train on all other chromosomes. So you'll notice that we're training on all other autosomal chromosomes except chromosome 14. You also notice that there's no chromosome 9 here. That's because Arrowhead has some sparsity issues when it comes with calling TADs on chromosome 9 at higher resolutions. That's another pitfall of conventional TAD callers, is that when you get to higher resolutions, you introduce sparsity in the contact matrix, which can throw off algorithms. So for consistency, we've omitted chromosome 9 here as well. Now, this is actually the function is pretty savvy. If you don't like that framework, if you prefer to build on chromosome 14 and also predict on chromosome 14, then the data is split into a 70-30 training testing ratio. Also, options, you could build on even chromosomes and predict on odd chromosomes if you prefer. You can do that as well. But we find this framework to be solid and allows you to, in the end, aggregate all the results and get a pretty solid average performance. Okay, so after you run this function, you can see what the data looks like. This function outputs a list of 2 objects. The first object is the training data. Is the training data, and we can see that by we can see the ratio of non-TAD boundary regions to TAD boundary regions is the same because we specified random undersampling here. And then you can evaluate the testing data, which is the second component. You'll notice at the top there, we're performing this model on 43 transcription factor binding sites, which it might not be necessary to build this random forest model on all of those transcription factor binding sites. So as a way to optimize our model to improve computational efficiency, we can take advantage of this TAD RFE function, which is a wrapper for the RFE function that's offered by the CREATE package. So essentially, we're taking our training data and we're building a random forest model recursively at different sets of predictors. So what this does is it builds a random forest at the maximum number of predictors, so 43 in our case. It identifies the top 32 predictors, rebuilds the model, and then recursively downward by powers of 2 essentially keeps building the model. And so what we see in this figure 3 here, also I just want to, sorry, I should have said this at the top, I just want to illustrate all the figures, the code for the figures are available here in this section, the PreciseTAD R package. All the code for to generate the figures are provided in the repository, which can be obtained from this embedded link here. So, okay, going back. So, what we see by evaluating essentially the cross-validated accuracy is that performance essentially converges when you only consider the top 8 most predictive features in this random forest. In our paper that's been submitted, we did this for across each holdout chromosome essentially, and so we were able to find that the most predictive influence was being carried by these top 4 genomic annotations. Now, in this case, this is only being done on chromosome 14, so on the x-axis is just each fold. In this case, we're performing 5-fold cross-validation. So each of these 8 predictors might not be the same across each cross-fold, so that's what this heat map here illustrates in panel B. So what we can see here is that really most of the predictive influence is being carried by these top 4 features, and so we can come to the conclusion that building a random forest model simply on these 4 is sufficient and can decrease our computational time by still maintaining that optimal performance. So to improve in that computational speed, we'll take our TAD data that we've already built, and we will restrict it to just include those top 4 transcription factor binding sites. And these are known in the literature that tells us our model is doing what it should be doing, essentially. CTCF RAD21, SMC3, and ZNF 143. So we want to restrict our training data and then our testing data to just these top 4. And now we can actually build our random forest model using that data. So we'll take advantage of the tadRandomForest function in preciseTAD. This is a wrapper for the random forest function provided in R. We specified the training and the testing data, the tuning parameters. The main tuning parameters for random forest are the number of variables to consider at each node when you're splitting, the number of trees, and then the minimum node size. We'll perform this using a 3-fold cross-validation. What metric we want to consider. There's multiple metrics, but in this case, we'll work with accuracy. The output of tadRandomForest includes a model object, which we'll need to use later for preciseTAD, whether to include the importances, and then some performance metrics when you take your model and you validate it on the test data that's provided inside the function. So we can see here, after running this, we have our performances again are stored as the 3rd object in the list, and then you can see kind of the performances which are listed here in table 1. So the first 4 components are basically elements of the confusion matrix: your true negatives, your false negatives, false positives, true positives. Again, a true negative is just a bin that was not a TAD boundary region, that was predicted as not being a TAD boundary region, and so on and so forth for each of these components, which are used to essentially calculate each of the following performance metrics here. Okay, so just want to keep going here. Okay, so I think I have some time to mention preciseTAD Hub. So all of this model building process can essentially be circumvented because we've already built these models and stored them on the cloud in the Experiment Hub, and that can be loaded in and read in using preciseTAD Hub, which is an Experiment Hub package that I mentioned before. The keynote is that you have to be working with R 4.1 or greater, or in this case is the development version of R. And you can read more about the naming convention of those files and how they're stored at your own pace. It's mentioned here, but it's also mentioned in the vignette associated with preciseTAD Hub. But essentially, each model has its own Experiment Hub ID that can be read in, and the naming convention is the chromosome that was used to be held out. This essentially is the chromosome you want to make predictions on. So a model here has been built on chromosomes 2 through 22, the cell line that was used, the resolution, and then the type of ground truth boundary. So in this case, it's Arrowhead. But we also have been working with chromatin loop boundaries, which unfortunately I don't have time to talk about today, but is an interesting, has interesting results. So those could be read in using this following command. If you're working with 4.1 or greater, first you have to load in the Experiment Hub library. So you have to do library Experiment Hub, initialize Experiment Hub, store the query and label the package name, and then specify the ID that you want to work with. So EH3863, 363. Yeah, so obviously, so we're working on chromosome 14, so that corresponds to this model. So we'd be reading in this model. So that's there for you, and that can save you some time if necessary. Okay. All right. So now we built our model, we optimized it. Now we can leverage that model using our main function preciseTAD. So again, we are the main function name is preciseTAD. So first, I want to make sure you understand, since we restricted our data matrix to only be those 4 transcription factors, we also have to make sure to restrict this GRangesList object that we created up top, which, remember, had all 43 bed files as a GRangesList. We need to go back and restrict that to just the top, to just these 4, which is done with this command here. And we will use this in our preciseTAD function. So the actual preciseTAD function has several parameters here. First is, again, that GRangesList object of your features. You'll use this to construct the base level feature space, that A matrix I talked about in the pseudocode. How you construct the associations, basically the feature type you use to construct that matrix A. Again, we want to use distance to match with how the model was built. The chromosome that you want to make predictions on, and then this chrome chords parameter is there if you just want to make predictions on specific section of the chromosome. So the default is null. In other words, you want to make predictions on the entire chromosome, but for the interest of this workshop, we're just going to be working on this specific region of chromosome 14 as an illustration. So this little over 1 megabase region. You provide the model object that you want to use. In our case, it's a random forest model, but it's up to the users. They can build whatever model using that CreateTagData function. They can build whatever model they prefer and store it here. And then the probability threshold that I mentioned before in the slides, whether or not you want to parallelize it, and then the dB scan parameters. You'll notice min points here set to the default of 3, and then the epsilon neighborhood we've used set as 10,000. The flanking is essentially, I'll talk about that later. We'll hold off if there's time, but that's not necessarily an important parameter for right now. So when you run this function, and you provide verbose equals true, it gives you a list of different things. So first, it tells you in this region that you provided, how many base pairs exceed your probability threshold of basically 1. So there's 29,000, essentially 29,000 potential boundaries of being, coordinates of being potential a TAD boundary. Now within each of those, preciseTAD has identified 8 clusters, and within each of those 8 clusters, it's identified the 8 predicted TAD boundaries. So the output of preciseTAD consists of a list. The primary components of the list are, again, that preciseTAD boundary regions, the genomic coordinates that span each cluster identified by dbScan in those 8 clusters, and then the respective boundary point within each cluster. And this is the boundaries that were predicted by preciseTAD that we're going to be comparing to Arrowhead within this region. Okay. So now I want to get into actually comparing the preciseTAD results in the last few minutes, because I want to leave a few minutes for questions. The way to compare boundaries are preciseTAD predicted boundaries with Arrowhead. First, you can load them into juicebox. Juicebox is a tool provided by the Aiden Lab, which essentially allows you to visualize TADs that were called or predicted on the contact matrix. So, so what we see here is this contact matrix is representative of the chromosomal coordinates that we specified in PreciseTAD. So in blue is what Arrowhead has called. You'll notice Arrowhead is actually inflating the number of boundaries here. Even in the small section, you see instances here, there's 2, here in their small sections 2, you know, so on and so forth. PreciseTAD, on the other hand, does not inflate the number of boundaries, and it also corresponds well with the intraconnectivity within the chromosomes, within the chromosome 14, for example. So you can see, just visually speaking, it's performing rather well. Now to get to the actual boundary comparison, I'll skip how we implemented, how we extract these enriched heat maps and profile plots. So on the top here in figure 6, you have the profile plot, and then on the bottom you have the enriched heat map. And this was carried out using deep tools. You can find more details in this section, techniques for visualizing TAD boundaries. But essentially what we see is that for chromosome 14, PreciseTAD, the PreciseTAD boundaries are much more enriched for CTCF sites compared to Arrowhead. And statistically speaking, they're actually shown to be statistically significantly closer to CTCFs for chromosome 14 than Arrowhead, which can be found in panel B here. And then lastly, we can see that PreciseTAD has higher levels of enrichment between 2 cell lines than Arrowhead does, which shows that PreciseTAD boundaries are more conserved across cell lines than Arrowhead. And again, these results are just illustrative of chromosome 14. I'll leave it there for now. There is a section on the cross-cell line prediction that I would like to get to, but I'd like to leave it for questions, unless I can continue if you all would like.
While people are waiting, let me ask, like, how do you envision the advantage of using PreciseTAD versus just calling TADs from, like, high resolution, high C data? From
well, so there's 2 benefits. One is the benefit that I didn't get to, unfortunately, cross-cell line prediction. And that's allows us to kind of, using this framework, to build predictive models on one cell line and then predict on another cell line that might not have high resolution data. Because it takes advantage of that conservation between cell lines. We've seen through our work, we've shown that this performs very accurately compared to same cell line prediction. The other benefit is, which kind of in line with the same point, is that, you know, performing this high C sequencing at high resolution is often time consuming, computationally expensive. And so taking advantage of this framework, you can build these models on low resolution ground truth boundaries and still maintain downstream precise prediction, which we have also shown in our paper. So something to look at as well. So those are kind of 2 benefits.
And perhaps I can add to it that you actually don't need high C data at all. You just need genomic annotations like CTCF, RAD21, and from them you can predict TAD boundaries without high C data at all. And for this you provide pre-trained models. Right,
exactly. Yeah, those pre-trained models, again, are provided in PreciseTAD Hub. Here. I don't like that it's showing that. Anyway.
All right, we don't have much other questions, but you're around and you can be contacted, I believe.
Sure.
Thanks very much, Mikhail and Spiro, for this great workshop.